I am here today, delighted to introduce you to my good friend, Professor Juliet Morrison at the University of California, Riverside. She trained with me when we were baby scientists, when we were trying to get our PhDs in Vincent Racaniello's lab at Columbia University. And Vincent is famous for being one of the great polio virologists. And that's actually what Juliet worked on in graduate school. I dabbled in polio, but I mostly worked with rhinovirus, which, believe it or not, is related to polio, although the common cold is not as bad as poliomyelitis. Juliet and I were talking, and we just thought that it would be a good way to catch up on some science that we love because the world is a terrible place today. And it's really nice to be able to actually talk about science. And it seems that polio is a topic that I think more people should know about, given that, unfortunately, I think there's going to be a lot more polio in all of our lives. So, without further ado, I'm going to let Juliet introduce herself briefly, and then we're just going to jump right into the greatest virus of all time to work with, not to have circulating around in the world.
Yes.
And I'm just going
Uh,
to sit here and drink my polio teeny.
Hello, audience. Yes, my name is Juliet Morrison. I'm a professor at University of California, Riverside. I study host responses to viral pathogens such as influenza virus and dengue virus, yellow fever virus. My original true love is polio virus, and I worked on that in Vincent Racaniello's lab. And yeah, Angie is completely correct. She is polio is like the best virus to work with. It is so robust in tissue culture, also very robust in the mouse model, and it's like the original gangster, like OG of viruses for learning about how our cells actually work. Because viruses require the cell to replicate. Studying viruses actually allows us to learn a lot about our own cellular physiology and cellular processes. So yeah, I is that enough of an introduction? Yeah, I
That's a
would say
great
let's go with Angie.
that's a that's a great introduction. You just like wrote like a grant abstract about polio from like 1975 or something. We need to find out about translational control.
Oh yeah.
Um, no, it's true. I mean, this is why I've been obsessed with polio lately, and this is kind of what gave me the idea when I was chatting with Juliet about it. I was thinking about our mentor Vincent, who had me on a show, and he likes my Substack. I think this is the first time ever in history. Juliet, would you say that Vincent likes me being outspoken and swearing a lot, and like, I'm doing it for a good cause these
Yeah,
days.
I think so.
So, it's been really great, like 25 years after grad school, reconnecting with my mentor like this. And so he asked me if I wanted to have a show that was based on my Substack where he would ask me questions and we just chat about, whatever I had written about. Usually what we're chatting about is the shit show that is the Department of Health and Human Services and all the terrible things that are happening to science and public health and vaccination right now. But anyways, I did so well that he ordered a full season of my show. And it just really occurred to me that just having a conversation with Vincent about viruses that don't have to prepare for, it's just a natural conversation about stuff that's going on with viruses between two people who've been studying them for their entire careers. And it's people seem to really like it. Like they seem to really are there seem to be learning a lot from watching those conversations. And so I thought, Juliet, we've always had great conversations. I mean, Juliet and I have been like not partners in crime because we're very law abiding people,
But yes,
but
we are.
we're just like partners, platonic life partners. We used to call ourselves, and we started
PLPs.
this. Yeah, PLPs. We started the same year in graduate school. And we really came up together. I was a couple years older than Juliet because I took a couple years off, and it's really funny to think of 21-year-old you calling 24-year-old me
Goldilocks.
Goldilocks because I was so old.
Yes, you're so old.
I mean,
I can't even
so
imagine. 23. Wow.
I never thought I'd be this old. Both of us are old now. But in any case, like we've been at this for a long time and like for the same amount of time. And we went through the same stuff in grad school, like studying more or less the same viruses. Both of us turned out to be good at different things, which is always fun. But in talking to Vincent, kind of reminiscing about all this stuff, I started refreshing myself with his old papers. And so, for people who aren't virologists, and even many virologists, Vincent was the first person to clone an animal virus. And he got really, really lucky. We could have another whole episode about that, talking
Yeah.
about that. But he was the first person to do it. So he invented essentially reverse genetics for animal viruses that like infect people and animals. And he did it with polio. And just like so much beautiful work has been done with polio. And I was going back over Vincent's old papers, and he discovered, or he was one of the groups that discovered the receptor for polio.
Mhm.
He developed one of the first transgenic mice for polio. He, as Juliet was saying, you can use viruses also to study things besides infection and pathogenesis, and you can actually learn a lot about just the way that cells work from them. And so another huge area that he worked on was translational control, or the way that cells regulate how protein gets made, which polio has a very interesting way of regulating, and that actually had a little bit to do with Juliet's
Yeah, a little bit. Yeah.
PhD research. Same protease. You were just studying a different aspect of it. Polio, like many pathogens, can antagonize the interferon system, and that's what Juliet studied with the 2A protease. I told you I would mention it. But anyways, we both really loved working with polio. And as Juliet said, it's so robust. I mean, you could leave that shit on the bench like overnight
Yeah.
and you would not have a reduction in titer. That's
Yeah, it is.
unfortunately one of the reasons why people could potentially pick up polio from the environment, because it's a really hardy virus. But God, what a beautiful
It,
virus.
it is incredibly stable. That is, I think, one of our biggest worries, that, it's sitting out there in sewage. And I mean, this is something that travels through our whole gastrointestinal system and manages to, infect our cells. That means it can survive at very um, big pH ranges that other viruses
Mhm.
cannot survive at. So, yeah, I'm worried that people are less interested in vaccination these days. Like, I think polio is going to make a comeback.
I think so. And you know what? It's corny, but like I've been saying this for months, like, and it's so far it's true. Like measles first, polio will be worse. Like I don't think people understand what a polio epidemic would be like in the US. I mean, when the first polio vaccine came out, the inactivated polio vaccine that Jonas Salk developed, it's just a whole virus inactivated vaccine, grow up a bunch of virus. Polio, it's easy. Jonas Salk did not have such an easy time with it because cell culture methods were not very good back then. And that was, of course, also before Henrietta Lacks was horribly exploited to really crank out the polio virus. But I got to say, her cells really do crank out the polio virus. Um, Jonas Salk was able to, partly with actually the help of HeLa cells, but he was able to grow poliovirus in cell culture, which was something that had been accomplished by John Enders, the developer of the measles vaccine, before. All those guys back then were like, they were all making vaccines, many of which are actually still used today, including both Jonas Salk and John Enders. Well, John Anders' measles virus was tweaked a little more by Maurice Hillebrand, who was also another big vaccinologist guy.
Mhm.
But, all these guys were like doing really fundamental virology. And I mean, John Enders won the Nobel Prize for what he did. Jonas Salk
And,
should
and,
have.
and, it, and they were considered heroes. I mean,
They
people
were.
were so happy that these guys were out there, like this is like these are movie stars, like
Yeah.
level quality, because they were saving lives, and I think
And
we've kind of
children.
forgotten that. And children, yeah,
Because I don't think people realize that when like you get poliomyelitis, like paralytic polio, it's game over for your whichever motor neurons are affected. So
yeah,
might be your legs. If it's not the entire leg and like you still got some that work, then great. You'll have like a limp and a polio leg. Um, you might be
or
fully
it could be,
paralyzed.
or it could be your diaphragm, and
It could be
there's
your
a
diaphragm.
lot. Yeah, exactly. And I
And
mean, we don't. Yeah, I don't even think we're even ready for that because we don't even. People are like, oh, isn't polio virus? Isn't polio virus eradicated? Is it? Do we even have a sense of the disease? Yes, we do. It's super, and the virus is super stable. So, and as long as there's international travel and the fact that a lot of communities now don't vaccinate in the U.S., like it's going to keep popping up.
yeah, and it's not just, I mean, at this point, I don't think it's just communities that are not vaccinating. There's going to be more and more people who are not vaccinating, not even because of policy changes. I mean, that obviously that's an area that I work in a lot, and that's a huge concern. Um, but at this point, like I don't think that Kennedy and the other people in charge of this whole disaster are going to actually ban vaccines. They're going to do everything possible to withhold access and make it difficult for people to get access to vaccines. However, I think that even the stuff that they fail at, and I mean, it has been like just throw the kitchen sink at the entire vaccination schedule and guidance from the CDC and see what sticks to limit access to vaccines. Even if it doesn't stick, it still reduces people's confidence in vaccines. And there's just relentless propaganda that you and me and all of our colleagues are all basically evil virologists who have been, I don't know, would you say that you're living large on the profits of big pharma, Juliet? Because
Oh,
I may be
the
drinking a
bus,
martini, but this is a glass that I bought from Target when I was a postdoc.
I take the bus to work, so like, I
I
mean,
mean, it's
and
silly.
vaccines don't make money for pharma anyway. Like this is something that has always had to be kind of like a public good. This is not something that makes anybody money. Like if you're thinking about a therapeutic, sure, but like a vaccine, like that's preventing the disease. So it's just conflating all of these things together that actually, if you really like were to, um, you know, use a bit of logic, you'd realize that it actually makes no sense. There is no way that we are academics are profiting from this, and even the pharmaceutical companies are not profiting from this. Well, maybe they did in COVID. I could talk about that a little bit, but in general, no. Like this is something that for the public good because we don't want kids dying, and we don't want to have the after effects that occur when people have get infectious diseases because there's always something else that it changes your immune system in general. It weakens things. Original antigenic sin is a thing. We can talk about
Do you
that
want to
another
explain?
time.
Do you want to explain like briefly though what that is? I don't think people know what that is, and like that is a concept that I think is really complicated, but it's
It is
really
super
interesting.
complicated.
But people hear original antigenic sin and they're like, "What? Like, what is
What
that?"
it's the idea that based on what you originally encounter as from a pathogen. So like pathogens will make proteins that are different from our proteins, and then the sites that are different from ours are we call them like epitopes, or and the epitopes make up antigens. And so if you are exposed to an antigen type from a virus, and then you are then exposed to another antigen from a related virus, but different enough that they're not quite the same virus, somehow your immune system will just keep making antibodies or have a response to the original virus instead of the second virus. So then you're actually less protected against your second virus. Is how I understand antigenic sin
Yeah.
originally to be. And so that's
I always
kind of
call it
yeah.
imprinting because I don't like to
Yes.
like mingle my science and my Catholicism. Um, but I mean, I think that that's why I don't mean to digress from the polio
Mm.
topic, but I feel
Oh
like
yeah, you can talk about whatever.
I also kind of want to brag to you, Julia. I mean, I love bragging to colleagues more than anything, especially in front of people. But yeah, I don't think this isn't even really something to brag about. It's like how I'm about to reveal how old I am. Um, Alexi, my husband, got influenza. I'm pretty sure. We didn't test. He just stayed home. It was not subclinical, but it was sub serious enough to go like try to get medical care for it. The time had passed for him to start taking oseltamivir. So I was like, just rest and like chill and stay away from me because I don't want to get flu either. Um, we both had flu shots. I checked. The year that I know that I was hospitalized for flu when I was a 1-year-old infant. Um, the dominant subtype that was circulating was H3N2. And right
Mm.
now, this dominant strain that is circulating is an H3N2 variant, essentially, that between the time that the strains were selected for the seasonal flu shots and now mutated, essentially, which happens periodically from flu season to flu season. It always happens because that's how influenza viruses work. And it's more vaccine evasive. It's a bad match this year. So I'm pretty sure that's what my husband got. I didn't get it because I think I committed original H3N2 antigenic sin when I was a year old in the hospital. And so I have better protection from my annual seasonal influenza vaccine, which is, by the way, the same technology that Jonas Salk used to make the polio vaccine. It is also the same technology that the NIH director, Jay Bhattacharya, is billing as a next generation universal flu vaccine platform,
Oh
which is
wow,
not.
really? That is not a next generation platform. That is
Wait a second, you didn't know about this, Juliet?
no, I did not know about this. I will
Okay,
tell you, I sometimes I just kind of turn the I just I check it on the news like maybe 3 times a week, and that's about it because otherwise I will go crazy. So
do you
I'm
want me? Do you want me to tell you about the corruption?
oh please do yes.
Okay, so in May Day, big communist Soviet holiday, according to my husband. Um, Jay Bhattacharya, Secretary Kennedy, I think Oz and Marty McCary, the FDA commissioner and CMS administrator, might have been there, announce a fantastic new initiative, Generation Gold Standard, the next generation innovative platform for a universal flu vaccine. I said, "What is this? Very interesting. Tell me more." As you know, Juliet, and as I'm about to tell everybody, it has been long a priority of the National Institute of Allergy and Infectious Diseases, or NIAID, Tony Fauci's former institute, to develop a universal flu vaccine. So this would be one shot, no more flu vaccines, one and done. Maybe you got to get an annual booster
Maybe a
because you
booster.
might need to boost those antibodies, but you don't need to get no strain selection, nothing. It works against all flu that you're going to be exposed to. The reason we don't have a shot like this is that this is actually an extremely challenging problem. And the traditional approaches that have been used for nearly 100 years have not worked for this so far. So, what is Generation Gold Standard? 100-year-old approach. It is a quadrivalent BPL inactivated whole virus flu vaccine that includes basically 4 recombinant reassortant avian low path strains that have been, reassorted with reverse genetics to express the broadest number of potential H's and N's that we have not yet been exposed to. So a bunch of random avian subtypes, basically. They did one study. Oh, Juliet, you're going to love all of this stuff.
Yeah, no, I'm like, I mean, I
They
can
did
see
one
what.
study, and you know who did it? You know who developed this? Jeffrey Taubenberger, the acting director of NIAID currently, and good friends and collaborators, longtime collaborator with Matt Memoli, the principal deputy director of NIH, who is essentially in like the good cop, bad cop like duo of Jay Bhattacharya and Bad Cop. He's the bad cop. He's the dude who cut like over 100 DEI grants in 6 minutes and then told a federal judge that he'd reviewed them all thoroughly.
That's
He
awful.
he's
Yeah.
he is a Maha minion. He is a very bad person. I have a lot on my Substack actually about the things that Matt Emily has done. But that's actually not the topic of this. The topic is still Generation Gold Standard. So Memoli developed this with Jeffrey Taubenberger. Jeffrey Taubenberger, as you may recall, Juliet, because you did your postdoc in his co-author's lab, um, reconstructed the 1918 influenza
Yeah,
virus.
I lecture on that all the time. I think students find that really exciting, this reconstruction of that virus, right?
Now that is called DGOF, dangerous gain of function research. So I guess it's okay because they already did it with 1918, but don't do it with anything else. But in any case, 1918 is a DGOF virus. So I look at this press release about Generation Gold Standard and I'm like, what the hell am I missing here? There must be some good fucking evidence about this. Because, like, to say that a whole virus inactivated vaccine, a quadrivalent, like, what
Sounds
is
so
it?
ridiculous. Like
What is it?
nothing different. It's
Where's
nothing
the
different for what? Yes. What's the
right?
innovation?
Where's the
Um,
evidence? So I look at the one paper that's been published on this. One paper. Science Translational Medicine, so great, but one paper. They tested this vaccine in mice and ferrets, and they tested it with fully reconstructed 1918 and 3 recombinant reassortants that had PB2 E627K inserted to make it more replication competent in a mammalian host. And then I looked up the paper how they chose these viruses.
what, gain of function work? What?
Not just hold up. It gets better on that note. I look up the paper that described how these viruses were generated. It was from like 15 years ago. And not only did they insert PB2 E627K, and for everybody watching, that is a mutation in avian influenza in the polymerase, the enzyme that copies the genome that makes it more fit or capable of replicating in a mammalian host. So they intentionally made this avian virus capable of replicating in a mammal. And then they made a bunch of reassortants where they stuck in a bunch of different avian HAs. So hemagglutinin, that is the protein on the surface of influenza virus, the H in the H5N1 or the H1N1. The H is what binds the receptor and allows the virus to get into the cell. And in bird flu versus human flu, that's very important because that determines whether or not it can transmit. It's one of the determinants. So they made a virus that was capable of better replication in mammalian cells once it got in there. And then they put on a bunch of these different HAs. And they selected the ones that were lethal in mice. So it was gain of function in mice. It doesn't necessarily mean that it would kill humans, but
It doesn't mean that, but I think
but they selected it based on the gain of function, and it's so fucking hypocritical.
Yeah,
And
it's crazy.
But this is the best part that you're going to really love, Juliet. And by really love, I mean you're going to absolutely hate it. You know how much they got for this? $500 million to develop this vaccine.
Oh my God. So they take old technology, they basically go against everything that the government has been saying we shouldn't do, and then they get $500 million. Okay, so I guess they spent like $100,000 setting up that experiment, and then where's the rest of that money? I would actually love to know because I, it doesn't. Okay, maybe like research is expensive. You have to pay your employees. So okay, let's say this was $500,000. How is this a $500 million project?
It's not
As
a
a
500.
taxpayer, I'm as a taxpayer, I'm actually quite upset because like what
It's it's
it's
it's just
ridiculous.
that's why I asked if you wanted to hear about the corruption. It's
Yeah,
corruption.
that's crazy.
And I've got other stuff on my Substack. Juliet, I was watching.
Okay, I need to read this stuff, but it also is going
Okay.
to depress me.
Oh, I know. You know me though. I try to keep it funny. I try to keep it light. But well, not light, but I'm like Freddy Krueger. I have like a gallows sense of humor. You
Right,
know,
no,
I like to
I
think
mean,
of myself actually as Freddy Krueger. And at some point I'm probably just gonna stick my head in like a barrel of fire and like I'll actually look like Freddy Krueger. But
I,
I'm I'm like
I'm sorry. I mean, dating yourself or something, but like, I don't even know who Freddy Krueger is.
the Nightmare on Elm. You've never seen a Nightmare on Elm Street?
Totally
Freddie? Like you've never seen Freddie, the guy with the knives?
I'm kidding. I'm kidding. It's like to make fun of you and your elderliness.
I fall for it every time. I've been doing this for fucking 25 years. We're gonna be in the old folks' home and you're gonna be like, who's Freddy Krueger? And I'm like, what do you mean you've never heard of Freddy Krueger thinking I'm so smart? It's always this way, Juliet.
I know.
Well, yeah, so this is the real problem. So before Christmas, I'll just tell you this story really quickly about continued corruption with a specific group of people. Um,
Oh,
I'm just scrolling the hell site, because I do this. It's like when you have you ever would you remember when you had like teeth fall out when you were a kid and like I couldn't help but like stick my tongue like sort of in the socket, like the tooth had fallen out of it. It kind of hurt, but it kind of was like satisfying at the same time.
totally
That's
satisfying.
kind of how I feel about like going on Twitter. Um, Anyways, so I'm on there and like, it's right before Christmas and like my world is falling apart for a variety of other reasons. And I see this video of Jay Bhattacharya and Matt Memoli doing the quote Pete and Bobby challenge. And the Pete and Bobby challenge is Secretary of War Pete Hegseth doing a bunch of push-ups and like pull-ups with uh Robert F. Kennedy Jr., who was wearing jeans the entire time. And this was like a few months ago. This is like I mean, in like internet viral trends, this is like geological time. But they did this right before Christmas, and it was the most like cringeworthy video you've ever seen. It's like Jay coming in to Matt Memoli's office being like, hey, Matt, like Christmas is coming. We got to get in shape. Got to take the Pete and Bobby challenge. And there's like, kind of guitar music. And like it says extreme for no reason. These are the two least extreme in the
Oh,
extreme sense kind of men,
I'm
but
so sorry.
extreme actually, I think, in the sort of fascist sense.
Oh, this is
In any case, like, so I watched this video and then I start making fun of them, but then I start kind of going down this rabbit hole about, this has always been kind of an unfinished nagging thought about Generation Gold Standard. And I remind you that this press release about it came out in May of last year. So I've had these months to like stew over it. And, I think about vaccines like all the time. I am the co-editor in chief of a journal called Vaccine. I read vaccine papers nonstop. I think about vaccines. I'm constantly defending vaccines. I do vaccine research. I work for a vaccine research institute. Like vaccines, vaccines, vaccines on the brain. Like it's like girls, 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 except vaccines, vaccines, vaccines for me. And I've been just thinking about this and it just really bugged me. Like that, and I've looked like these are the only papers about Generation Gold Standard. But I don't know why it didn't occur to me to do this. It's to look up what the fuck Matt Memely actually does. You know what he does? I know what Jeffrey Taubenberger does. His contributions as a virologist have been exceptional, and I want to note that. Bringing back 1918 has contributed immeasurably to our understanding of influenza and our ability to prevent influenza pandemics. It has made it easier for us to select vaccine strains. It has done so much for public health. He has advanced the science of virology. And with influenza, that's a competitive field. As you know, Juliet, you're more of a flu person than I am. I dabble.
Very, very competitive.
I'm a host person. That's why I get to go to all the parties and people like me because they don't see me as a competitor. Flu's like a cutthroat ass field, and I'm not even trying to be like one of those flu people.
Believe me, I don't even consider myself. I mean, like I am more now at this point like an innate immunologist who loves viruses, but flu is just one. It's one. It's a great virus, though. I mean, a great group of viruses. I mean, talk about like ridiculously difficult to work with. But the fact that there's so many different strains of it, and the fact that so many H's, so many N's, so much your assortment. You could go at it for days, but at the same time, you're telling me that somebody got you know funding for technology that is not new and is basically from the days of old. I mean, like Noah and his ark were like around at that point. Like that sounds kind of ridiculous. At the same
The thing
time,
is,
though, I'll go back to this to say that we still don't quite understand what determines vaccine efficacy. So, like the fact that these people are like, okay, let's just pick some random stuff and let's throw it at the wall. Like, I mean, maybe it will stick is something, but it doesn't sound like something that should require that to necessitate that level of funding. Like that sounds crazy.
it's an insane level of funding for that. And you know what the basis, I mean, we're going to just get into the weeds. And like I said, we're just going to have a conversation and people can pick up some virology. Yeah. But they did a heterosubtypic challenge in this paper. And so for everybody watching, what that means is that they immunize these mice against H, 2 and, well, that's a human subtype, so I shouldn't say that. H10N6, I don't remember what it was, some avian subtype that they made up. So they immunize them against that, and they have this collection of H's and N's. And I have it in a Substack. I don't remember off the top of my head exactly what they all were, but it was like H1, H3, H5, and H7, I think. And then it was no, it wasn't H7. They didn't have H7 in it. That was one of the heterosubtypic challenges. They claimed that it was going to be universal because they basically stuck this quadrivalent vaccine in that has 4 H's and 4 different N's. And they stuck in viruses that had either one different H or N, and then they stuck one that was, no H's or N's in common, and it protected mice against all of them. And it provided some protection against ferrets, but they didn't do that much ferret work. So protecting mice, it's a start. I do a lot of mouse work. I'm not knocking the mouse model at all. But you're talking about putting $500 million into a single mouse study and saying that this is going to, on the basis of using basically a virus that you made up to see if this can cross subtypes. I mean, this seasonal flu is a great example. We've all been exposed multiple times to H3N2. It's one of the main seasonal flu strains that's been circulating since 19 whatever, before I was born, um, in the late 70s. Um, it was circulating before that. I actually never meant to tell you the other part of that story. My husband is older than me. He was probably imprinted with H2N2, which is probably why he got sick and I didn't. Finish that story. Anyways, um, a heterosubtypic challenge in a mouse model like this does not prove that you have a universal flu vaccine. This is encouraging preclinical data. It is not something you award $500 million for with no scientific review. Because if there had been a scientific review, you know that they would not have accepted a single fucking paper, even if it was in the science journal,
Yeah,
not science.
no, it just doesn't make any sense. It's wow.
It's corruption. But anyways, so I start looking into Matt Memley. And Matt Memley is doing human challenge studies. He started a human challenge study cohort and he has multiple papers. I have an entire table and appendix at the end of this Substack article, like it's the supplementary material in a fucking journal. Because he has written so many papers in which he has deliberately infected human volunteers with viruses that he made from reverse genetic clones, and in some cases selected vaccine evasive mutants from, and in some cases then cloned and reinfected new human volunteers with. So that's the work that he's doing. Now, is that dangerous? It's not, because he's working with seasonal flu. But the fact is, he is still doing work that he is saying is unbelievably dangerous because he canceled the grant of a guy named Christopher Brook at the University of Illinois Urbana-Champaign, who studied seasonal flu evolution and selected in cell culture for mutants that would evade antibodies induced by the vaccines. He did this because A, it's not dangerous. These are seasonal flu strains, and a vaccine evasive flu strain is next year's flu strain. It's like what flu does normally. Still dangerous viruses, but they're circulating out in the world. This is not dangerous gain of function research that wouldn't result in viruses that are everywhere anyways in the human population. The reason he's doing it is to find how the virus is going to evolve under this type of selection pressure in the actual human population that he's trying to make better flu vaccines for. So if we understand better what flu is going to do, we could predict what strains the vaccine should be much more accurately.
Right, yeah.
That grant was canceled. That grant was canceled because Matt Memley's research competes with it. He's doing the same thing that he said was dangerous, except instead of doing it in cell culture, he's doing it in human volunteers at the NIH Clinical Center.
Insane.
It's insanely hypocritical.
I mean, it's insanely hypocritical, but like if you
And it's
really
insane.
if you were to really worry about these things, which I worry less about these things, but if your idea about transmission and the creation of a new strain that might wipe out humanity or whatever, like zombie apocalypse that you're thinking about, um, putting it in human volunteers is actually like kind of the like that's the that's the movie. That's the movie. It's not the cell culture experiment is not the movie. This is I can't like
Juliet, so, you know, maybe we can, maybe this is a good way to get back to the polio vaccine because what, what's interesting about now and then, there are so many different things, and I'm so fascinated with history. And as you know, because Juliet knows me, like I've always been really into history. But I'm so interested in the history of the polio vaccines because of something you said earlier. They were heroes. I mean, Jonas Salk, who not everybody thinks that Jonas Salk is incredibly hot, but I sure do because he's totally my type. Like an extremely educated man who is a workaholic and like doesn't want any money for it. Like
Oh,
he's my guy. Um, he was also kind of a hoe. So I like him also because of that. So so his biography said. Anyways, Jonas Salk was amazing. And he didn't want any money for the polio vaccine. People asked him if he was going to patent it, and he said, "Could you patent the sun?" I mean, like that is an
so
attitude.
badass. That is so
It's so
badass. Like,
badass. Like it
right.
is just like heroic. It is absolutely heroic. And
Yeah.
they were rightfully hailed as heroes. And I'm not saying that we and all of our colleagues, who are working at this should be hailed as heroes.
No.
But what I'm saying is that like Jonas Salk was hailed as a hero not because of that. He was hailed as a hero because he developed the polio vaccine and within a year of releasing that vaccine, I mean, there was a reduction in thousands of cases of paralyzed children. It was just incredible. And that vaccine, and I was going to talk about this, but we're just, this is it. I'm not as stringent as Vincent, so I'll just order another season right now. We'll come back and talk more about how the vaccine works. But as it turns out, the polio vaccine is not a sterilizing vaccine. Jonas Salk couldn't have known that. He wasn't able to test that vaccine the same way that we are able to test it now. He was able to grow the vaccine, make the vaccine, and basically immediately start clinical trials. So anti vaxxers now like to talk about these, historical experiments that were really done in a different world. They could barely grow the virus. This is something that for us, I mean, when we were so lucky to learn how to do virology with polio because it is such an easy virus to work with.
Agreed.
It is
Agreed.
it is elementary to work with. You have to try to fuck up like working with polio. Contaminate your HeLa cells, do something, but you really have to
Yeah.
you really can't screw it up. You can't even screw up the reverse genetic system. It's not even normal transfection. I have described this to my students, and they're like, "What do you mean DEAE dextran transfection?" And I'm like, "Yeah, we didn't have lipo. Vincent did not have lipofectamine 3000. I mean,
Yeah.
the reagents, the standards for doing research, for doing clinical trials, for developing vaccines are completely different now than they were back then. And the urgency. I mean, this is what kills me about the entire vaccine question is that back then the urgency was so great. So many kids were dying. The year before the Salk vaccine came out was a peak year for polio. Thousands of children died from polio.
Yeah.
Thousands more were paralyzed and spent the rest of their lives in an iron lung or disabled. Um, the majority of people who get polio infections don't even know that they have it. But for the people that get poliomyelitis, when it gets out of your gut and into your motor neurons, like you're fucked forever. And if that happens to you when you're 6, like how is that something that is okay and natural if we can prevent it?
If we can, exactly, like it makes no sense to me. Like, why would we be able to come up with experiments, come up with ideas, come up with therapeutics, come up with vaccines if it wasn't a natural thing to do? Like, let's just do this so that people don't have to suffer. I No, I think I think it's so unethical to bargain with people's lives, and especially with people when the science literacy is so low at this point that people don't actually know, what it should be. Like it's unfortunate. Like it makes me sad.
Do you remember? I know you didn't do that. You didn't do as much pathogenesis work as I did in graduate school, but do you remember, like, I know that you saw paralyzed mice because we did that
Yeah.
collaboration together.
Mhm.
And the polio control mice in that experiment. I mean, do you remember what that looked like? Because I'll never forget when I was training, and another one of Vincent's graduate students trained me to work with the polio mice. And I worked with him to do a couple experiments to get used to handling the mice. And I'll just never forget what it looked like to see a mouse with polio for the first time. It was
Yeah.
honestly, it's one of the most horrible phenotypes I've ever seen in terms of so a phenotype is like when you infect an animal or cells or whatever with a virus and then something happens. Whatever happens, that's the phenotype. A disease phenotype is what that disease looks like. And so for poliomyelitis, that's flaccid hind limb paralysis. And it's not just that the mouse is dragging its leg. Like I've seen paralyzed or disabled animals.
Really sad.
It's shaking. It has encephalitis. I mean,
Mhm.
it's, and it doesn't matter. The first time I saw it, we infected the mice intracranially. So that's sticking a needle basically into their skull. And I said that this is not remotely comparable to like a normal route of infection. So I started doing all of mine intraperitoneally. And that's a whole other episode again. Like we don't want to get into it right now, but it's hard to infect polio mice via the oral route, which is the natural route, because of a quirk in the circumstances of the gut in lab mice. And we should talk about that another time because that's also a really interesting story. And that's actually how I got my PhD was Julie Pfeiffer's seminal discovery about that. Anyways, polio, has I hate it when I do this. I got so distracted by that little soliloquy about Julie Pfeiffer's experiment. Polio causes this encephalitis, whether you infect by the intracranial route or the intraperitoneal route. Um, if you infect intraperitoneally, which is basically injecting straight into the peritoneal cavity, which is the main cavity in your abdomen, um, it will eventually find its way into the motor neurons of the spinal cord and cause the mice to get just as paralyzed and just as sick as if you stuck the virus straight into its brain. So it it doesn't really matter. Polio is a horrible virus. And after seeing that, even in a mouse, I never imagined that I would be in a situation where I'm explaining to Americans what polio is in the context of preparing them to see it happen to their fucking children. It is
Yeah,
appalling to me.
it is. Like
It is
I
the most appalling thing that has ever happened, and it is so immoral. I just,
it's not that
I try to get mad about the scientific part of it, but what have we been doing the scientific part for if not to prevent what they're gonna bring back intentionally?
yeah, no, it's really this is I I have no words actually. I
That's why I invented the polio
but
teeny.
because
It's just a martini, by the way.
we don't even have like the safety nets that are gonna like help people when their kids are like disabled from polio and all of this stuff. Like I just like
Juliet,
it's
it's
just
all
so much of everything. I'm so tired. But yeah, guys who are listening, don't like like the polio vaccine. Actually, what we really should be focusing on, I would say, is that we need to try to figure out how to like actually get rid of. We need to eradicate polio for real.
mhm.
And yes, there is a better oral polio vaccine available now. But I think personally that what we really need to be doing is, and I'm not a public health person, but just based on my understanding of virology, we need to be using the inactivated vaccine at this point in all of the hotspots so that we are not selecting anymore for these like vaccine um variants. Yeah.
Yeah, the vaccine-derived polio. I
Yes.
mean, this is a
Exactly.
this is the thing I hear
I
the
know,
most.
and I know it's more expensive, but like at this point, we know how stable the virus is. So if we're doing live attenuated viruses, even if they're actually better than the previous ones, I think the better investment would be to just have inactivated polio vaccines and move forward from there. I But that's what I lecture when I talk to my students. I'm like, for me, somebody would have to convince me otherwise that there's a better strategy than that. Like we need to stop putting out infectious virus. But at the same time, it's still better than not vaccinating because that's even worse. Like that's so much worse. So I'm not trying to say that listeners like we need to vaccinate, but I think we need to if we are so close to potentially eradicating polio, like we should focus on making sure that we don't introduce like vaccine derived polio into the environment.
I mean, do you think that so the vaccine-derived polio, at least for type 2, is the main circulating, like, and it's the main cause of paralysis, but that's because, again, they released, like, a oral polio vaccine that didn't include the type 2 component because the wild type 2 had been eradicated, but people didn't appreciate at the time that there are some people who can get infected with polio and shed it for long periods of time as well. So not only does it persist in the environment, but there are people walking around who have no idea that they're just like excreting literally a shit ton of polio every time they take a shit. And
Yep.
and it's very, again, as Juliet was saying, it's very, very stable. It's a huge problem. And, I mean, I've been out of the coronavirus world for quite a while, but now I have to get back into it, of course, because of my work with vaccines. I was done with these goddamn viruses. Like, they're not even enveloped. They're so annoying. And they're also difficult, except for polio. Polio is the only good one. I mean, it's not good. It's the worst of them. But well, maybe foot and mouth disease might run a close second. But
Yeah, that's a good one.
that
Yeah.
it's an agricultural pathogen. But I mean, hepatitis A, whatever. We can all the big ones we can conquer with vaccines, except for the rhinoviruses. But there's so many of them. I should maybe ask for $500 million to dust off my graduate research, develop a universal rhinovirus vaccine.
I think you should.
I can
No,
reboot that mouse model. I was just a couple passages away from getting that shit to like really replicate in mice.
I mean, I guess.
I'm gonna throw myself into the sun if I ever have to work with rhinovirus 1A again. Um, so no, that sadly, guys, like
I feel like rhinovirus 14 though would have been the one to go for. Like that one seemed to be the
16.
closest. Oh, it was 16. Okay,
It was
16. That was the
16.
one that was
16L was the jam. Oh no, rhinovirus 14 was the closest to polio. It was
yeah,
the weird
exactly. Like
one.
it was like really good at replicating. So
And it still didn't replicate very well in mice, though.
yeah.
Not in mice. I still have to do the same shit. Ooh, DGOF, serial passage in mouse cells. This is my favorite part. And one of these days I am going to write like a detailed Substack about just the stupidity of my PhD thesis
Mhm.
and how I did a fuck ton of what would now be called DGOF to basically make a mouse that would, or a virus that would grow in mice, but still didn't make them sick. Probably wouldn't have transmitted between them. It was like,
Oh no, you're frozen. Oh, Yanji. Um, hello? Hmm. God, let's see here. Um, Hmm. Not really sure how this works. Um, Oh no, it seems like Angie's phone died. I don't know. Are people still no, no, I'm not connected anymore. Um, Honey Badger, can you hear me? Let's see if we can get Angie back on. Let's plug her phone in. Okay. All right. Let me text with her. Well, actually, her phone's dead. But I assume she has her iPad. I will email her. All right. Give me a second. Let me.
Okay, hi everybody. Oops. I my phone died and I am just coming back on to say goodnight actually once Juliet returns. I apologize for that. I am quite bad at Substack live technical management and it may appear that I am drunk. However, I am not. I am quite incompetent and I don't quite know what's happening. Okay, guys. This is probably your indication to start, signing off, going to bed, getting dinner. I don't even know if anybody can see me because it appears that my camera's not working, or at least I can't see myself. So I am not sure when Juliet's going to be back. I'm waiting for her, but I'm going to begin the process of saying goodnight. So next time, Juliet and I have one of these conversations, it is going to be with
I'm back.
me
I'm back.
having. Oh, hi, Julie Poe.
Hi.
I my camera, I am not figuring out how
I
to work.
understand. Yeah.
I think it's time to sign off, but I was just about to say to all of our very patient viewers that we should do this again. We should continue this conversation. We are taking requests as far as what you'd like us to chat about.
Yes.
And next time, I promise to have my phone fully charged so that this does not occur again. Because whatever I was saying before I was shocked into, holy shit, my phone just died, was very profound and I forgot what it was. So we have to chat again and discuss that. Anyways,
Yeah. I mean,
Juliet,
no, thank you for having me on, Angie. This was really fun.
dude,
I
I
always love
think,
talking about viruses with you. And yeah, let's do it again.
yeah, let's do it like in, week after next, or we can do it
All right.
whenever. I mean, this is
Yeah.
actually quite easy to set up. So if you feel like chatting about something, just be like, let's do it.
Yeah, I would really like to hear what the audience wants to hear about. Like what's like, what floats your boat? Like, do you want to talk about H5N1? Do you want to talk about Mpox. Like, let's talk about stuff. I'm down.
It's true. And I have to say, Juliet is also an expert in macrophages. So she's kind of like me in that she doesn't really care what virus it is. And most of them, if they don't infect macrophages directly, like my favorite viruses, the hemorrhagic fever viruses usually do,
Mhm.
then they certainly have a lot of effects on macrophages. So Juliet's always got something to contribute to the conversation, just like me coming in hot with my little transcriptomic profiles. So,
Let's do
anyways,
it.
this was wonderful, Juliet. Thank you so much for coming
And
here.
and and thank you. This was fun. All right,
And
let's
thank you,
talk
everybody, for
more.
watching. I'm going to be a better Substack live host going forward. Hit like or subscribe or whatever. I don't even care about that. I'm just glad that you watched and stuck around for this. And next week, or next time, 2 weeks maybe, we'll
2 weeks.
yeah, we'll have a signature cocktail. I'm going to have a signature cocktail.
I
And we
will
should
come up
plan
with
that.
one as well. Okay.
People
All
like
right.
that idea. I see hearts. All right.
Yes.
Good night, everybody. Thank you so
All right.
much. Good night, Julia.
Toodles, y'all. All right. Bye, guys.
Honey.
Bye.